Welcome all. Welcome to the From Grapes to Wine School Climate webinars. My name is Neslihan Ivit. I'm the Winemaking and Innovation Specialist with Perennia. And today our co-host is Francisco Diez. He's the Viticulture Specialist. And today we are honored to host Dr. Vivien Measday. She will be talking about identification of 4 distinct Saccharomyces cerevisiae wine yeast populations from British Columbia's Okanagan wine region. Before we begin, I will give some Zoom housekeeping items. So you will have your audio and video turned off during the webinar, but we do encourage you to ask questions. And for that, please use the Q&A feature at the bottom of your screen. And Francisco will be monitoring the Q&A during the session. And after our presentation, he will be addressing your questions to our speaker. An introduction to Perennia. Perennia is a provincial agency, and Perennia's vision is that Nova Scotia is a recognized world leader in producing innovative, environmentally responsible, safe food of impeccable quality. Perennia's mission is supporting growth, transformation, and economic development in Nova Scotia's agriculture, seafood, food and beverage sectors. Our service areas is extensive. We offer services in agriculture, quality and food safety, product development. We have lab services, as well as we serve to fisheries. And when we deeper look into wine site, we offer services from vine to wine. Those are site assessments, viticulture services. We have a plant health lab. We offer winemaking services, mobile bottling and filtration, and industry development. Today's webinar is part of our industry development efforts. And today's session looks deeply into yeast populations. Yeasts are important for winemaking. Of course, as we all know, they transform grape juice into wine, but they also contribute a lot to other properties of the wine, like its color, aromas, profile, and mouthfeel. So they have a direct impact on the profile and the quality of the wines. And when it comes to applying them in alcoholic fermentation, we have 2 main strategies. The first one is the inoculation. In the inoculation, we use the commercial yeast starters or active dry yeasts. And those are generally Saccharomyces cerevisiae and its different strains. But nowadays in the market, we also have non-conventional yeasts. Those can be non-Saccharomyces yeast, such as Torulaspora, Meshnikovia, or Schizosaccharomyces pombe, a lot of different genera. But also there are Saccharomyces non-cerevisiae yeasts, such as S. uvarum. So what we can do is we can choose one of them, or we can even mix them with co-inoculation in order to achieve our desired quality and specific style of wine. And on the other hand, the second strategy is the spontaneous one. In the spontaneous one, we have uninoculated fermentations, and those fermentations are carried out with indigenous yeast. They are also known as wild, natural, or autochthonous yeast. And those yeast during the fermentation, they actually have a progressive growth pattern, and the population changes during the process of fermentation. And at the end of the fermentation, normally we have again Saccharomyces cerevisiae, who is finishing up our fermentation. But where do these yeast coming? That's the question. Of course, we carry them with grapes from the vineyard, but we do also have it inside of the winery environment and on the winery equipment. And since this type of yeasts are used frequently, there have been a question to identify what are those yeast populations and to study in specific wine regions and in geographical areas. And one of those studies is done in British Columbia by Dr. Vivien Measday. So we are very happy to have her today. She kindly accepted our invitation. Dr. Vivien Measday has a PhD from University of Toronto, and she have a postdoc research in yeast chromosome segregation from University of British Columbia. And in 2004, she started as assistant professor in the Wine Research Center in UBC. And since 2012, she has been the associate professor in the same faculty. And Dr. Vivien Measday study looks deeper in the yeast population in vineyards of British Columbia. With that, I will leave the floor to Vivien, and please join me welcoming her.
Well, thank you very much for the introduction and the invitation to present my work, Nestle, and also thanks to Perennia and Francisco for monitoring the questions. And I will just share my screen now. Okay, can you see this?
Yes, I think this is the PDF version, Vivien.
No, this is the PowerPoint.
Okay.
Is it not showing up as a? Oh, you're right. I'm sorry, you're right. It's the PDF version. Hang on a second. Sorry about that. Let me just
no
see
problem.
the right one. Okay. Well, the PDF wouldn't be so bad. Okay, we'll start again here.
Perfect.
Okay, is that right?
Yes, thank you.
Okay, thanks. I mean, I've only been doing Zoom for 2 years now, so I don't know why it's still a problem sometimes. Okay, so thanks to everyone who's listening today. And I'm going to talk about the work that my lab has done at the Wine Research Center at the University of British Columbia in Vancouver, British Columbia, with the wine region, and I'll explain in a minute where it is, called the Okanagan Valley. And this is some of the scenery that we can get in the Okanagan Valley wine region with the vines above the lake, Okanagan Lake here. Is very beautiful, and the next slide will show you a bit more about where exactly it is. Okay, so this is, can you see my pointer here? Maybe I'll go to laser pointer. Okay, so here is Vancouver, where university is situated in the lab, and then in the other colors are the wine regions of British Columbia. They start over here on Vancouver Island with about 32 wineries, and these numbers may be a little off, and then you can see the number of acres planted and some of the main varietals. And these little islands here are called the Gulf Islands. There's 7 wineries in total there. The surrounding Vancouver region, and including Abbotsford, that's where we had all the flooding, unfortunately, a while ago. That's the Fraser Valley, where there's about 25 wineries, but the majority of the wineries are in this region here known as the Okanagan Valley, and close to it, the Smilkameen Valley. This is about, depending on how fast you drive, I'd say about 4 to 5 hour drive from Vancouver to get over here. And the Okanagan Valley stretches all the way from up here in the north, Salmon Arm, all the way down to Osoyoos. So that's about 250 kilometers. They are approximately 185 wineries. It could be a bit higher now. And it represents about 84% of the total vineyard acreage in BC is in this region here. What's very interesting about the Okanagan Valley is the climate change throughout this valley. So the north is a lot cooler than the south. The Osoyoos is actually like a semi-desert with temperatures that can get up to 40 degrees during the day, but cool off at night, which makes the grapes in this region interesting because they can maintain their acidity due to the cooling off at night. So we've been working with wineries in this region, mostly between Kelowna and Osoyoos, in particular a winery in Summerland called Okanagan Crush Pad, and a winery in Oliver called Stoneboat Vineyards. And so what I'd like to talk about today is the microflora that's in the vineyard. And everybody's heard of the term terroir and how the terroir can really impact the wine that you drink, especially the same varietal grown in different regions can taste very different. And this is a combination of a lot of factors, including the terrain that the vines are grown on, the soil, the climate, how the wine is actually made. And this is where we come in, which is the microflora. So as Nestle mentioned, it is common practice these days for wineries to inoculate with a commercial strain of the yeast Saccharomyces cerevisiae, which is the yeast that converts the sugars to ethanol and also imparts flavor and aroma compounds. There are wineries who do spontaneous fermentations, which means, or they're called indigenous or natural fermentations, which is no inoculation. And that relies on the microflora on the grapes, on the winery equipment, etc. And it has been shown that by some studies that these microflora do change depending on the region. So what my lab's been interested in for the last 6 to 8 years is identifying the microflora, more specifically Saccharomyces cerevisiae, although we have done some isolation of non-Saccharomyces yeast as well. But we're interested in looking at the microflora in the vineyards and the wineries. And so we've done this using 2 different ways. So we've collaborated with wineries such as Okanagan Crush Pad, Stoneboat Vineyards, Tinhorn Creek Vineyards, and others, but the first 2 are the primary ones we've worked with. We've collaborated with them so that they've performed spontaneous fermentations in their winery and then sent us samples for yeast isolation. And when we convince a winery to do this, if we want to publish this work, we have to ask them to do the fermentations in triplicate, which is not always easy to do the same juice in 3 large batches in a tank. So we really appreciate that these wineries have been willing to do this with us. We can also isolate other ways, but when it comes to trying to publish the work, that's when we kind of need to do things in triplicates. The other way we've done it is to go into the vineyards associated with the wineries and take the grapes, and we ask permission. We don't go and take them without permission. Take the grapes and take them straight back to the lab, and then do the spontaneous fermentations in much smaller scale in our lab and isolate the yeast. The reason we've done this is because we know that every winery we've worked with also uses commercial strains, because spontaneous fermentations are quite risky, of course. They can get stuck if Saccharomyces cerevisiae maybe doesn't predominate. And so most wineries do a combination of commercial inoculation and spontaneous fermentation. And we know that those commercial strains, even if you don't inoculate them into a tank, are still present in the winery, and I'll show you some data from that. So that's why we also wanted to take the grapes from the vineyards and take them directly back to the lab so they didn't go near the winery equipment. Okay, so I'm going to show you an example of one of the studies that we did. And this was quite an ambitious study that my former student, Jay Martiniak, set up to do, where for 2 years we sampled from 13 different Pinot Noir vineyards, from the north in Kelowna to the south in Osoyoos, and every circle here is a vineyard that we sampled from. So there was 4 vineyards in Kelowna, 5 in the Summerland and Naramata Penticton region, and 4 in the Oliver Osoyoos region. And so what Jay and another graduate student, Elaine Cheng, did was for 2 years, they went before harvest. So this also relies on the winery telling us when they're going to harvest, so we get the grapes before they're picked. Just before harvest, we meaning my students, have to drive out 5 hours to the Okanagan Valley, sample the grapes and get them back to the lab. And you can imagine not all these wineries harvest at the same time, so that was quite a lot of back and forth. And so this is an example of how we sampled the vineyards. So what we did in this case, because every vineyard, of course, is different. When you've got 13 different vineyards, they're all different sizes and organizations. So we picked a fairly small region, a 0.25 hectare section of the vineyard, because that was a size that fit into every single vineyard we sampled, which worked out to about 16 rows by 12 post panels. And then Jay set up a program just to randomly sample these areas. So what's in red here would be the region of the vineyard that we sampled. So for each of these clusters, berry clusters were picked. And in order that we don't touch them and add our own yeast, whatever's on our hands, into the sample, we do everything with gloves on and we sterilize. Jay actually brings like a lighter and ethanol into the vineyard. I don't know if we actually told the vineyard managers that, but anyways, so we make sure everything's sterile so we don't add our own flora to the grapes. We put them in sterile bags and we take them back to the lab, which again is a 5-hour drive away. And then what we do for each vineyard is we set up 6 different fermentations. So these each represent those 6 different regions of the vineyard. And we crush them manually. So the berries stay in that bag and they get manually crushed. And then we use sterile lab equipment to transfer the grape juice into flasks. And we put them in the incubator at 25 degrees. And the way this is what they look like. The way we monitor them is by weight loss, because when the sugars get converted to ethanol, there's carbon dioxide release. And that results in actual weight loss. So we can tell if it's bubbling as well, but we can tell if it's progressing by measuring it on a scale, which is quite useful, at least for the lab. In total, we did, for those 13 vineyards, that was a total of 78 of these 500 ml flasks. And we did that for 2 years. And when we did this experiment, we had already had some practice with sampling, and we knew that by about 60% sugar depletion was good timing to isolate the yeast. We have done other studies where we've sampled throughout the fermentation. And usually if you sample earlier on, you're less likely to there's going to be a larger diversity of other yeast species. So it just depends on what you're looking for as to when you want to sample, and also how many fermentations you have and how many plates you want to plate out, et cetera, et cetera. So when we plate them, this is an example here of what the plates look like. And we have to do different dilutions because we're not sure exactly what dilution will be the best to get the colonies to grow. We also have to include certain drugs in the plates to make sure that bacteria and mold don't grow. So if we just put them onto this media with no selection whatsoever, it would just be a big mess of bacteria, mold, and yeast. And so we're interested in the yeast isolation. So we have to add some compounds to the plates just so we focus on the yeast. And so then my students spend a lot of time picking colonies, and I don't know if you can see, but there's different sizes and different things going on these plates. So they have to pick carefully, and sometimes we can tell just by looking at the plate if it's a different type of yeast, like a non-Saccharomyces. And we have been saving those as well. So we have a selection media, we call it Wallerstein agar, that we use to separate out what are Saccharomyces species from non-Saccharomyces. And basically it's just a green dye that for whatever reason the Saccharomyces are able to metabolize, and so they convert it, they get rid of it, and they're cream colored. And if it's not a Saccharomyces species, it can't do it. So these are green. So if it's green, we save it in a different part of the freezer than these cream colored ones. So after these studies, we have accumulated literally thousands of what we call yeast isolates. So when they come directly off these plates from a fermentation, we call it an isolate. The reason for that is it's quite possible that, you know, these two colonies here are exactly the same strain. We don't know yet because we haven't looked carefully. But when we say strain, we're really now talking about genetics. So we're talking about what is going on at the DNA level and how similar are they. And if something is called the same strain, it means it's very similar or the same genetically to each other, or they could be very different. And so the next part of what I'm going to talk about is how do we figure out what is the same, what is different, and importantly, what is a commercial or not a commercial strain. So in order to do this in a high throughput manner, because we have so many colonies that we've isolated, we put them onto these plates that are called omni trays, and we can fit 96 colonies into these at a time. And what we are next doing is a procedure that's essentially can be thought of as like fingerprinting. So everybody's heard of DNA fingerprinting for crime scenes if they're trying to match somebody's blood sample or DNA. And so this is kind of what we're doing here, although there was no crime scene. What we're doing is we're extracting DNA. So these are the yeast colonies. We're breaking them apart and we're doing DNA extraction. And then we're looking at specific regions of the genome of the yeast strains. These are repetitive regions that helps us to distinguish whether a strain is the same as another one or different. And so it's 10 different regions that get amplified using a method called polymerase chain reaction. And then what we're interested in is looking at the size of these regions. And so a strain that has, for all these 10 regions or loci, we call genetic loci, if they're exactly the same as each other, then we call it the same strain essentially. So this is similar to actually for people in academia, similar to how Sanger sequencing is done, where you're just measuring the size of each fragment using capillary electrophoresis. And then we use software to analyze the size of each locus. Okay, so this is a very messy diagram, and I'm going to try and explain it so that everybody can understand. So what we've done here is we've taken a lot of different of these isolates, and what we're doing is we're comparing them to commercial strains. So that microsatellite analysis or fingerprinting analysis that I just talked about, we also have done that for a lot of commercial strains. So in this hairball diagram, all the commercial strains are in red. And so anytime a strain is near another one, it means it's very similar to it. So like Fermentation or Anchor Vin 11, Fort or Rosé, etc. So any strain that's right next to another one, it means it's genetically similar. So what we were excited about is we found populations of our strains. So our strains, the ones we've isolated, we just colored them according to what region of the valley they came from. So the north is Kelowna, Penticton Naramata is in the middle, and Oliver Soyuz is at the bottom. And so you can see we've got strains here that have been isolated from both Oliver Soyuz and Kelowna that are quite similar to each other. And there's no red here, so that means there's no commercial strains that look the same. And down here we have a cluster from Kelowna of strains that look like they're potentially unique. And we weren't too sure about these two up here. So there's a few conclusions from this study. The one was that there was populations that were not the same as commercial strains, and that in general, often strains from a certain region would be more similar to each other than from another region, although there is some overlap, but more of the Oliver Soyuz strains cluster together and more of the Kelowna strains cluster together. So this is just to show you how much variation we can see in a vineyard. So what you're looking at here is a pie chart of the strains that were identified in a certain block of the vineyard. So this vineyard, we were fortunate all 6 fermentations worked, and we isolated yeast from all of them. And so, for example, the clusters we got from this part of the vineyard turned out to be mostly commercial strains. So the red hues here are commercial strains, whereas over here, and this is not far away, we got green, which green means non-commercial. So genetically they look different. Over here in this part of the vineyard, we got this fermentation was dominated by 1 commercial strain. And these ones are a mixture. So you can see just, it really does depend on where you sample from and how you do the sampling as to what kind of results you might get. So this is an example of a vineyard where all the fermentations worked. You can also get this scenario. This scenario here means 5 out of the 6, we got nothing. So that means the fermentations just didn't proceed. There wasn't enough cerevisiae there to complete, except for this one region here, which was dominated by a commercial strain. So for anyone thinking of doing these analysis in general, I recommend doing, well, I have a few recommendations that I can tell you at the end if you're interested. So this was just an overall summary of that particular study, the 2-year study we did with the Pinot Noir sampling. In total, we identified, based on microsatellite analysis, 109 Saccharomyces strains. 42 of these turned out to be commercial. 61 were unknown, so not commercial. 6 actually turned out to be Saccharomyces eubarum, which surprised us. We have isolated Saccharomyces eubarum from Pinot Gris, and it's a cryotolerant Saccharomyces species and is usually isolated in spontaneous fermentations of white wines, not red, so that was a bit unexpected. On the left here shows you the genotypes or the strains by region. The main point of this slide, this diagram here, is to show how little overlap there is between regions. So for example, in Oliver Soyuz, we got 32 unique strains. In Kelowna, we got 24. In Penticton and Naramata, we got 3. And there's no strain that was found in all 3 locations. There was only 2 strains, and I'm talking about the unknown strains here, that were found in both Oliver and Soyuz and Kelowna. The other thing on the right here that's interesting is there's very little overlap between strains by vintage. So, and we're not the only researchers to have discovered this, that you can see in 2016, in total, we got 39 strains, and I'm just focusing on the unknowns here. In 2017, we got 19, and there was only 3 in total that overlapped between both years. And so just to mention the commercial quickly, so 32 different commercial strains in 2016, 5 in 2017 and 5 that overlapped. And we only found the Saccharomyces eubarum in the second year. So there's definitely change between the years. The strains don't seem to stick around forever in the vineyard, let's put it that way. And so this is another study, so it's not the same as the one I just talked about. But in this one, what we've done is this is also Pinot Noir, and we've compared a winery fermentation to a fermentation done in the lab. And in this case, it's exactly the same vineyard that the grapes were sourced from that went into the winery versus that we took back to the lab. So in red are the commercial strains, white. So this was a few years ago, and we were calling them similar to commercial, but for all purposes, you could consider these the same. And then green is different from commercial. And here's an example where we did take samples throughout the fermentation, so early, mid, and late fermentation. And the overall conclusion is, even if you take the same grapes and do a spontaneous fermentation in the winery versus going back to the lab and doing a smaller scale spontaneous fermentation, you do see more commercial strains. So that would just be, if you're looking at the red and the white combined, you see more commercial strains in the spontaneous winery fermentations, which is really not surprising because we know that these strains are on the, even though these tanks are cleaned and everything's been rinsed, the strains do persist in the winery. And so in the lab, there was a bit of variability. So one of the fermentations clearly had a lot of commercial strains and the next two didn't. And so this is when I say like the results are often quite heterogeneous, you just really do not know what to expect when you do spontaneous fermentations. And so this is a summary of what we have frozen down in my freezer back in the lab from all the studies we've done for the past 6 to 8 years. So this is Okanagan Crush Pad Winery in Summerland that has been very, very supportive of my research. We have about 150 strains or genotypes that are not different from commercial. This is Stoneboat Vineyards, which my student, Jay Martiniak, his parents actually owned this, which is in part why we were able to get in there and do triplicate fermentations. And we found about the same number of strains that are not commercial from this vineyard and winery. We've also done microsatellite analysis on 160 commercial wine strains. And that's so we can compare, because of course, if we don't have a commercial wine strain in our database, then we don't know if a strain we've isolated is similar to it or not. So we have a total of 160, and I think there's probably maybe around 200 available. So we're actually pretty close to having all of the commercial strains genotyped. And finally, we have sampled from other vineyards and wineries in the Okanagan Valley. So we have 100 to 200 strains from other vineyards and wineries that are not commercial. So at this point, we know that we have unique strains, but all we've done so far is this DNA fingerprinting, which is a very useful indicator, but it's not the same as looking at the entire genome. And so the only way to really be 100% sure that what you're looking at is a non-commercial strain that is potentially indigenous to a region is to do whole genome sequencing. And there's a lot of yeast researchers out there right now interested in whole genome sequencing of Saccharomyces cerevisiae strains, not just from wine, but from other industrial processes like beer, bakery, sake, etc., which is good because it means we have other data to compare our data to. So I was able to get funding to do whole genome sequencing of 75 Saccharomyces cerevisiae strains. It would have been great to be able to do all of them, but of course this is expensive work. So we had to choose 75. And this was a critical choice because I didn't want to choose 75 and have them all end up being very similar to each other. So what you're going to see now is another one of these complicated diagrams with a lot of strains overlapping each other. And so excuse the mess here, but what the point here is to show just how many strains we have. So I don't know if I mentioned this before, but when you're looking at these diagrams, every word here represents a different strain. This strain on the left here, S288C, happens to be a lab strain that we frequently use for different type of work, using yeast as a model system. And so we've colored them based on what they are. So once again, the red is commercial. And in this case, we've colored them based on what we chose for sequencing. So if it's in black, it means we didn't pick that strain, because strains that are near each other, like I said before, are likely very similar. So we didn't want to pick strains that were all of the strains that were right next to each other, because we wanted some diversity in our sequencing. And so this circle up here represents strains that we were convinced by our microsatellite analysis turned out to would be different from commercial, but we also had to choose strains that were clustering near the commercial strains just to make sure, and some strains that were kind of on their own here. So in total, we picked 75 of these. And this is just an overview of the sequencing data for any academics out there. We did this in the Michael Smith Genome Sciences Center, which is a well-known sequencing center. We just isolated the DNA, they did everything else, and they did a great job. I did pay them for it, but I still thank them for doing a great job. This is done on a Lumina HiSeq platform with 150 base paired end reads. I won't go on through all this, but we have about 205 fold coverage. And what we're looking at when we are looking at these strains after we get all this data, we have to compare them to what we call a reference genome. And this reference genome is our lab genome. It's the first eukaryotic genome ever sequenced, called S288C. It's very well annotated. And what we find is most of our reads, sequencing reads, map back to this genome, which means it's very similar to this one. But there's what we call SNPs, or single nucleotide polymorphisms, that are nucleotide changes. And that's what we, and by we, I don't mean me, I mean people that do bioinformatics work, look at to see how different are these strains from each other. Okay, so this is a summary of the sequencing data. And so this is a phylogenetic tree. And every time you see a circle here, it represents a yeast strain that we sequenced. And what we did is we compared our strains to some global strains. So we included strains on the left here are isolates from China. So yeast actually originates from oak trees, if you can believe it. And so these are wild strains that were picked from trees in primordial or primeval forests in China. Then we have some sake commercial strains. We have a strain isolated from a North American oak tree, and we included commercial wine strains in our analysis. So we were really excited to find that we had 4 different populations of yeast. And so what we found was a lot of our yeast, all the ones in the red here, are similar to commercial wine strains, and we expected this. In fact, I was a bit concerned that they would all look like this, but we definitely expected this, and so this is what we found. And anytime a strain is close to another strain, it means it's similar. So all these ones in red essentially are genetically similar to each other. We're excited by this group here, the Okanagan Valley wine, because they don't seem to be similar to any other strain we've looked at. And then on the next few slides, I'm going to explain these ones in a bit more detail. Okay, so like I just said, Saccharomyces cerevisiae is actually isolated. You can isolate it from natural habitat, and it happens to be an oak tree that's probably the best place to isolate what we call wild cerevisiae. So in order for it to be called wild, it has to be isolated from a tree that's nowhere near industrial or human contact. So because we're trying to isolate what was out there in nature. And it's been shown not by my group, but by another group, that the commercial yeast are similar to a yeast strain isolated from an oak tree in the Mediterranean. Okay, so before I get into that, what we've got here are sake yeast strains. So both of these two here are commercial sake strains that are used for sake production. And we were surprised to find that two of our strains, these were the ones actually isolated in Naramata and Penticton region, were genetically similar to sake. That, I believe, is the first time that's been found, that a strain isolated from wine juice is this similar to a sake strain. So the next thing we found is that we've got some of our Okanagan strains that are actually similar to a strain isolated from a North American oak tree. And I'm going to show one possibility for why this is in a minute. And we were really excited by this because another group has shown that the commercial strains of yeast are similar to this strain here, which was isolated from a Mediterranean oak tree. And so what their proposal is that all the commercial wine strains have essentially been, what's the right word here, have their parent or their progenitor strain comes from this oak tree, and they've been domesticated from this oak tree into wine production. And you can see that a lot of the commercial strains are genetically very, very similar to each other. Okay, so I just wanted to explain a little bit more about this oak strain. This strain here, YJM1273, was actually isolated from an oak tree in the United States, and not anywhere near us, but way over here in Pennsylvania. Actually closer to you if you're joining from Nova Scotia. So you might think, how on earth can we be isolating strains all the way over here in the Okanagan Valley that are genetically similar to a strain isolated from an oak tree in Pennsylvania? And although I have no evidence whatsoever for this, one possibility is that this is one of the regions in the United States that makes oak barrels, wine oak barrels, whiskey oak barrels, other types of oak barrels. So one theory is that when you get your oak barrel from wherever you get it from, there could possibly be yeast associated with the wood in that barrel, and that's how it could have arrived over in the Okanagan. There's lots of other potential explanations too, but this is a nice one because it's a nice story. So that's what I'm going to say. And I think I already said this, that for the green strains, which we're calling the Okanagan Valley cluster, we have not found what we call a global strain. We have not identified any global strain so far that's genetically similar to these, which is exciting because we can consider these to be indigenous to the Okanagan Valley. And we're also considering the North American oak as indigenous. Basically anything that's not related to a commercial strain, we're considering as potentially an indigenous strain. So for the last couple of minutes here, What I wanted to talk about is how do we go from all this screening and genetic data to making something useful out of this for the wine industry. And what we're starting to do now in collaboration with Okanagan Crush Pad is to look at strains that we isolated from their vineyards and winery as potentially potential starter cultures. And not just necessarily for their winery, but potential starter cultures that we could commercialize. And so I just wanted to give a big thanks to Okanagan Crush Pad owners, Christine Coletta and Steve Lorney. Christine has been extremely supportive of my research, and without their funding, I couldn't have done a lot of this work. And Matt Dumaine, their winemaker, who's been very helpful with providing advice, providing juice. So we really do rely on wineries to participate in our research programs. And in this case, I've, in order to do the starter culture development, I had to get funding as well. So I'd like to thank Investment Agriculture Foundation of BC, who also helped fund the sequencing project. And then my tax for helping me to pay for a student. I recruited Elia. She's originally from Mexico and she has experience in the tequila industry, and she's now doing a food science degree in my lab at UBC. And so she's just started. So we've really just started this culture development work. But what we first need to do is we need to figure out if these strains that we've identified, can they actually work by themselves? Because when we identified them from these spontaneous fermentations, there was a lot of strains all mixed together and other yeast species as well. And we don't know unless we try whether when we inoculate them individually, what will happen. So what Elia is going to be doing is we've picked all the strains we consider to be indigenous that are Okanagan Crush Pad strains, one that's similar to the North American oak strain, and 5 that are in that Okanagan Valley cluster. And we always include a commercial control because we're comparing to what we want. We want a strain that we know can complete fermentation. And we've chosen to test these out in 2 varietals that are very predominant in the Okanagan Valley, and that of course Okanagan Crush Pad is interested in Pinot Gris and Pinot Noir. And Matt very kindly sent us large tubs of frozen juice so that we can do our experiments all year round. And the next choice is, well, what temperature do we use? And so we've tried a few different temperatures. We obviously want a temperature that the winery will be interested in, but also we want to maximize our ability to detect aroma compounds. So for the Pinot Gris, Elia's done the 15 degrees and has just almost completed the 20 degrees. And for Pinot Noir, we still have to pick the final temperatures. So after we figure out what strains can actually complete the fermentations, the next thing is to do slightly more robust, larger scale fermentations and look very carefully at metabolites. So for metabolites, we look at soluble metabolites like the sugars, acids, ethanol, and then we look at volatile metabolites. And so this is gas chromatography, mass spec. This is high performance liquid chromatography for the solubles. So for the volatile metabolites, we're really looking at aroma compounds, esters, terpenes, etc. Okay, this is my last data slide here. So this is work that Elia has done recently with a fermentation and Pinot Gris juice sent to us by Okanagan Crush Pad at a very low temperature, so 15 degrees. And we're looking at just residual sugar here. So this is residual glucose is in the blue and fructose is in the orange. So this strain on the far left here did not ferment at all. So there's 100% residual sugar. I believe this one has done a lot better at 20 degrees. So 15 degrees was too cold for it. On the far right here is the commercial strain. So we just do them in different batches for extra controls. And that one has consumed all the glucose and left a bit of residual fructose. The other strains are not doing too badly. They're not quite as robust as the commercial one, but we don't necessarily expect that. But they have managed to consume the majority of the glucose, and they do have some residual fructose. I think these values will look different when we go to 20 degrees. I think part of this was that the fermentations were a bit too cool. So far, I'm actually quite happy that 5 out of the 6 were able to ferment at 15 degrees, because we just didn't know until we tried which ones would be able to ferment at all. Okay, and I see that it's 10 to 11, so I just want to acknowledge a lot of different people here. I'm going to start with the funding agencies, because without the funding, we can't do the research. NSERC, which is the federal agency for the natural sciences and engineering research council, for funding my lab, the British Columbia Wine and Grape Council, and I already mentioned Investment Agriculture Foundation of BC and MyTax. There's a lot of people in my lab who contributed to this work. Especially the sequencing analysis was done by Alex and Jackson. Jay and Jonah were involved in getting the strains ready and off to the sequencing center, and Elaine was involved in isolating strains, and Elia's working on the starter cultures. The wineries, of course, that have been so supportive of the work. I hope they continue to support me. And I didn't get into a lot of the bioinformatics work because I didn't think it would be that interesting for everybody. But the sequencing analysis required us to collaborate with a bioinformatics company, and Arya Han has been the point person there. He's been very helpful, and finally the sequencing center. And I'd like to thank everyone for joining, and I hope you have some questions for me. So thank you.
Thank you very much, Vivien. It has been a great presentation. It's such a deep study, taking all different aspects of wine, like viticulture is there, genetic is there. It's truly amazing. Thank you very much for sharing it.
You're welcome.
And Francisco, can we pass to the Q&A part?
Absolutely. So Vivien, we have a question, and they are asking, is the yeast competing with each other during fermentation when there are multiple strains fermenting together?
Yes, definitely. And I do have other slides showing that information, but I didn't, I should have showed it today, but I didn't, sorry. So yeah, what we generally see is in the beginning of the fermentation, we see different yeast species predominating. We don't actually see Saccharomyces cerevisiae until a bit later on. So it depends on how we do the study, but it definitely progresses over time to predominantly being Saccharomyces cerevisiae at the end. And even amongst the different Saccharomyces cerevisiae strains, they also can compete with each other. So some strains have something called killer toxin that they secrete, where they can kill off every other strain there. And we actually found that a long time ago in one of the tanks at actually Okanagan Crush Pad. Was one tank was predominated by a strain that had killer toxin. So, and that was something that wasn't inoculated. So they definitely can compete. And I think one thing we're interested in doing when I get more students and money is to try to blend these strains together and see how they work with each other. Because in fact, they may produce better wine if there's more than one of them. We just don't know yet. Like there's quite a bit of research that needs to be done.
Perfect. And I have a couple of questions to you, Vivien, taking the opportunity. My first question is, do you have or did you see any difference between the vineyards? I mean, which was the management of each vineyard? Conventional, organic, biodynamic? It exists anything like that?
So definitely some of the vineyards, for example, Okanagan Crush Pad is an organic vineyard. Some were organic and some were more conventionally farmed. We didn't have any overall conclusions. Like it wasn't like we only got yeast from the organic farms or we only got them from the conventional. We did, for example, notice that in one region, the Naramata-Penticton area, that we, for whatever reason, got less yeast. And we actually think that might be more to do with the way the wind flows. So if you can imagine, something's got to move these yeast from the winery onto the vineyards. And it could be something as simple as wind direction or where the vineyards are located relevant in relation to the winery. But I mean, I think generally speaking, you might be more likely to find yeast if you don't, spray a lot of antifungals. And I do know that Jay was able to tell, so his dad runs the vineyards for Stoneboat Vineyards, and he did ask him to not spray too much. But of course, we can't go and ask. We don't want to go to every vineyard that we're working with and tell them not to spray because they have to make wine. So, but yeah, so I guess the conclusion is we don't have any conclusions. There are other people who publish studies on that though. But yeah, we definitely did in the case of one vineyard anyways, ask them not to overdo it with the antifungal sprays.
Perfect. And my last question, you show the vineyard 11 in Kelowna, where you have some issues. So you said you were going to give some recommendations. So I'm very curious about that.
Okay, so the recommendations are more for how to isolate yeast from vineyards. And so we've done it two different ways. So we did it the way which I showed, where we take 5 separate regions and isolate and then do separate fermentations. The way I would say that was the most lucrative as far as getting strains was to take samples throughout the entire vineyard. So not to do like 3 small sections at a time, but to sample the entire vineyard 3 times, which is so across the whole vineyard. And then instead of taking just from a small region and putting it into a flask, taking like 6. So instead, I would say take 6 groups of yeast from across the vineyard, put that in a flask and do the same thing again for replicates. So sample as wide a source of the vineyard as you can, because if you restrict yourself to small regions here and there, you can get what I just showed, which is nothing, which is disappointing. I mean, we were okay because we did 13 vineyards and 78 fermentations, but I would say half of them, less than half 1 year worked. So if you're only doing 1 vineyard, it'd be really disappointing to sample and get nothing, which in some vineyards, that's what happened because of, so how you sample does make a big difference. So anyways, if anyone is interested, they can always email me for a bit better scientific but a bit better explanation of how I would recommend doing it.
Thank you very much. And Nestle, if you would like to ask a question, I don't want to monopolize this section.
Sure. So Vivien, you explained it so easy. Almost we were going to believe that it's such a, you explained so well that it sounds like easy, but we know that it's such a deep study with all the sterile sampling and the triplicate fermentations. So I wanted to ask you, what was the most challenging part of all these activities?
Well, I think when we first started, Jay was really determined that we do everything in the same day. So it's more it wasn't me, it was the students, but they would go out to the Okanagan, stay the night, sample in the morning, and sometimes 3 or 4 vineyards, and then bring everything back to the lab, and we would do all the crushing on the same day. So I did have some slides of us starting the crush at 9 o'clock at night, and we didn't leave the lab till 3 in the morning. And that was with about 6 or 7 people working. So after that, we said, okay, we're not going to completely exhaust ourselves. And we actually decided to put the grapes overnight in the 4 degrees, and it didn't really make much difference. So you don't actually have to stay up all night. But on the other hand, it made us more aware of what it's like to work in a winery when you do have to keep going, right? So one thing that is challenging for us is the distance, obviously, between Vancouver and the wineries. I should say our wine research center is both campuses. We have a campus in Vancouver and a campus in Kelowna. And so we do have researchers on both campuses. And obviously, if you're in Kelowna, it's a lot easier to physically get to the vineyard or the winery.
Perfect. Thank you very much, Vivien, for answering all the questions and presenting us today.
Okay, thanks for inviting me.
I would like to thank you all, the attendees, for joining us today, and I would like to invite you to our next session. In our next session, it will be January 24, next Monday. We will have Dr. Marie du Toit from University of Stellenbosch in South Africa, and she will be talking about cool climate malolactic fermentation and what does it mean in the year 2022. So we are waiting all of you there on Monday. And finally, if you are CCA, CPAG, CPSS, and CPSC, I would invite you to make a scan of this QR. I will give 30 seconds for you to make the scan of this code. Perfect. Thank you very much for attending.